Second speaker today is Dr. Marcel Goldschen-Ohm, Associate Professor in the Department of Neuroscience at the University of Texas at Austin. Marcel is internationally recognized for his contributions to ion channel biophysics, structure-function relationships, and mechanistic modeling of synaptic receptors. He earned his BS in physics from St. Cloud University in 2001 and went on to complete his PhD in 2009 in physics and biophysics at the University of Wisconsin-Madison, where he developed an expertise in patch clamp electrophysiology and Markov modeling of GABA receptor gating. He remained at Wisconsin for his postdoctoral training with Baron Chanda, where he built a uniquely interdisciplinary skill set by collaborating with leaders in single molecule photonics, ion channel pharmacology, and snare biology, training that now defines his mechanistic sophistication in his lab. Since establishing his independent laboratory at UT Austin in 2017, Marcel has led a research program focused on understanding the molecular energetics of ligand-gated ion channels. His group is known for using genetic code expansion to interrogate main chain chemistry and probing atomistic contributions to gating that are inaccessible with traditional mutagenesis alone. A wonderful example of this, which I think he may be talking about today, is a recent Nature Communications paper that was published this year, where he and collaborators used genetic code expansion and Xenopus oocytes to incorporate alpha hydroxy amino acids into the GABA receptor, revealing that a single backbone interaction contributes roughly a third of the energy required to open the channel. This is a super exciting result that reframes longstanding assumptions about where gating energetics originate. Marcel has published over 25 papers, is funded by an R1, and is simply put among the top modern ion channel biophysicists. So we are thrilled to have you today. Marcel, please take it away.
Thanks so much for that wonderful introduction. Yeah, so we heard about some sort of amazing sort of tool building. And here what I'm going to show you is use of a sort of long-standing tool, although very underutilized in my opinion, for ion channels, and show you what we've learned biophysically about the protein from it. So quick background. I'm going to tell you about GABAA receptors. So GABAA receptors are neurotransmitter-gated ion channels, and they are in postsynaptic cells. And so when you have an action potential arrive at a presynaptic site, you have vesicles filled with neurotransmitter or GABA. They release GABA into the cleft, binds to postsynaptic receptors. They open up, and chloride flux through these receptors then inhibits the postsynaptic cell from firing its action potential, or at least generally so. So these are essentially the transistors in inhibitory synapses throughout the brain. And not surprisingly, there are all sorts of diseases related to variants in these channels. You can make a lot of different GABA receptors. There are 19, I think, different subunits, and you can form in different combinations. Today, I'm going to tell you only about the very typical synaptic subtype, which is going to be an alpha, 1, beta, and gamma 2 subunit. So very simply, this is sort of a simple view of how they work. At rest, the channel is closed. GABA, or neurotransmitter binding, binds to extracellular sites, which opens the ion pore and allows chloride flux. And then they rapidly go to a desensitized conformation, which is due to closure of a secondary gate closer to the intracellular side of the membrane. And here you can just see some representative current traces from either a brief pulse of GABA or a prolonged pulse of GABA, where you can see this desensitization while GABA is around. So we have a wealth of cryo-EM structures now for these synaptic heteromeric subtype GABA receptors, so alpha, beta, gamma. So here we have the resting conformation, where people have looked at primarily in the presence of antagonists, which hold the channel closed. This is a bit of an old slide. We actually do have some activated conformations now, but for a long time, this was an issue. And the reason it was hard to get these is because when you put on neurotransmitter, the channel rapidly desensitizes. So all the structures in complex with with activators were desensitized. So recently, Radu Arăcescu's lab has published a whole bunch of some very cool and interesting structures where they've done flash freezing after spraying the receptor with GABA for only a few, I think 10 milliseconds. And this is still in probably in peer review, but the bioRxiv paper is available. The structures are available. I'm not really going to go into a lot of detail about this here, but we do at least have some ideas about activated states. Okay, so this is one of the older structures, I believe from Ryan Hibb's lab. And here you have, it's a pentamer, so there are two alpha subunits, two beta subunits, one gamma subunit. Here we're looking down from the top of the receptor. You can see GABA or neurotransmitter is bound at the beta alpha interfaces. So that's two binding sites for neurotransmitter. In this particular subtype, there's also a benzodiazepine binding site between alpha and gamma subunits. If we look at from the side, the extracellular domains are these sort of beta barrel regions. Here's where you bind neurotransmitter or benzodiazepines. And then the transmembrane domains here contain the main gates, both for the activation and desensitization gates. And what I'm going to focus on today is, I think, so still, even though we have now some structures of presumably activated channels, what is still, in my opinion, something that is not well understood. And that is, how do these sort of loops right at the interface couple the binding of neurotransmitter to gating of the pore? And in particular, I'm going to start out by focusing on this one loop here shown in panel B. This is the M2-3 linker. So M2 is the pore lining helix. M3 is the next helix. And this linker is known to be important for gating. So certainly mutations in this, as well as other loops in this region, can have big effects on channel function. But there is still no real comprehensive view at the molecular level as exactly what interactions are contributing how much, and sort of what is the overall detailed functional interactions that matter here at this interface. And this is what we're going to at least look at one piece of. And so here was the paper referred to. So in part one, I'm going to tell you about this recent publication. And then in part two, I'll tell you about some unpublished work. So when we looked at these structures, what we noticed was this M2-M3 linker adopts this sort of very similar structural motif across all subunits, and in fact across all members of the superfamily cis loop pentameric ligand-gated ion channels. And so that includes nicotinic acetylcholine receptors, glycine receptors, serotonin receptors. And so here you have the pore lining helix, and you have this M2-M3 linker look like it adopts almost kind of like a W shape. So it's got these sort of two lobes, and then it enters into the M3 helix. And when we looked at this, we thought perhaps the shape of this is very important for gating of the receptor, and it appeared that potentially it could be stabilized by hydrogen bonding between atoms in the backbone itself. And so, for example, one of these was this proline, the carbonyl oxygen, and then the nitrogen, the amide nitrogen from this isoleucine. These bonds, I want to point out in the structures, are not necessarily well defined. So if you look at different structures, sometimes if you use default hydrogen bond sort of cutoff criteria in programs like Chimera X, sometimes you'll see them, sometimes they won't be identified. Either the atoms will be slightly too far apart or the angles will be slightly too mismatched. And so the structures don't really well resolve whether or not this bond exists or not. Nonetheless, they show that these atoms are in relatively close proximity. And so we thought maybe there is a bond. How can we interrogate this? So obviously, we can't directly interrogate this bond using conventional mutagenesis, as all subunits are going to have the exact same backbone. I suppose we could put a proline residue here, which would eliminate hydrogen bonding to this nitrogen, but proline introduces kinks, and so that could introduce a lot of other aspects that we don't really want. What we wanted to do was specifically disrupt this bond without changing anything else. And for that, we turn to non-canonical amino acids, where we're going to specifically change this amide bond to an ester bond. So essentially swap a nitrogen for an oxygen in order to ablate these bonds specifically without changing anything else about the side chains. And to do that, we collaborated with Chris Ahern and Jason Galpin in his lab, who can synthesize these alpha hydroxy acids. So here's an example just for isoleucine, but a similar alpha hydroxy can be made for a number of other amino acid residues. So here you have the amide nitrogen with isoleucine. It can hydrogen bond to the carbonyl of the backbone of another residue. And if we replace this with this alpha hydroxy acid with the ester bond, we have essentially ablated that hydrogen bond without altering the side chain. So how do we introduce these? We are going to do electrophysiology in Xenopus laevis oocytes. So these are eggs from frogs. And the way that we do this is we introduce a stop codon. And so this is going to be a typical nonsense suppression method. And then we co-inject the RNA for the GABA receptor subunits containing this stop codon wherever we want to introduce the unnatural with tRNAs that have been ligated to the unnatural, which would be the alpha hydroxy version of whatever residue we're replacing. And we also do this exact same procedure, but using tRNAs that are ligated to the wild type amino acid as a control. So this condition should produce wild type proteins, so it should behave as normal. And as a secondary control, we inject with tRNA not ligated to any amino acid. So this should leave a stop codon in the protein. It should be truncated and not express. So here we should see no currents unless we have some sort of read-through at the stop position. And so here's the basic experiment. We take the oocytes from Xenopus laevis frogs, inject them with mRNA, including stop codon plus tRNAs for each of those three conditions, and then record in 2-electrode voltage clamp the current through the channels in response to application of, for example, GABA. So here is sort of what a typical experiment would look like for this particular paper. Is our protocol was to interrogate the channel with picrotoxin. So this blocks the pore of the channel. And the reason to do this is if the channel is spontaneously active, so open even in the absence of neurotransmitter, then we'll see a reduction in current. So upwards is a reduction, downwards is increased current. So this block will be an assay for whether or not the channel is spontaneously active. Following that, we deliver a dose response in GABA and then another picrotoxin pulse just to assay rundown over the course of the experiment. And the main takeaway for this in these bottom graphs is that we are able to successfully introduce the either the wild type, so this is at a position in alpha 1 in that M2M3 linker, where we reintroduce the wild type valine using the nonsense suppression, or we introduce the alpha hydroxy version as compared to the background. So we get nice currents in all conditions, but when we have the blank, so this should be a stop codon, we see no currents, so no expression. So this is successful, good introduction with little to no read-through. And the same is true. We did the same thing in the beta 2 subunit at the analogous position. And so here we introduced the wild type isoleucine or the isoleucine alpha hydroxy. And again, we get nice expression and nothing when we have the blank. And same thing, so this last one, I'll just mention, when we first started this, we expected that the energetic effects of these of ablating these hydrogen bonds might be very small. So we started out by doing this actually in a gain-of-function mutant, and that mutant has a mutation in the pore gate, which causes it to be spontaneously active. And so that's actually what you're looking at here. And that's why we can see this blocked by pyriprotoxin, because this gain-of-function mutant is just active all the time, about 20 to 30%. And so we started out in this gain-of-function mutant. However, the effects we saw were quite big, so we ended up doing the same thing in wild type. And so here you're just seeing in wild type, same thing. We do take an expression hit compared to wild type when we do the nonsense suppression, but we still get resolvable currents, whereas we see nothing in the blank. So here is the main result. We're starting off in the alpha subunit, breaking that hydrogen bond with the alpha hydroxy, or just recapitulating wild type. And we're doing this in this gain of function background, which has some spontaneous activity. So here's reintroduction of the wild type. It looks just like normal gain of function. And if we then introduce the alpha hydroxy, there is no change. So we still see about the same amount of spontaneous activity compared to full activation in GABA. However, if we do this in the beta subunit, well, we start off with the wild type control. It looks just like gain of function wild type. But if we introduce the alpha hydroxy, so we break that hydrogen bond in the linker and beta subunit, we get this massive increase in spontaneous activity in the channel. And this increase caused us to suspect that the effect was much larger than we thought it might be, and hence explore it in the wild type background. And so here now in a wild type background, so in a wild type, the channel is not open at rest. And so we apply pyriprotoxin, there's no effect. That's because the channel is fully closed. But when we break that hydrogen bond in the beta subunit using the alpha hydroxy for isoleucine, then we see actually some spontaneous activity. Now, this looks like it might just be a very small effect. We're only getting a little bit of spontaneous activity. However, this is a bit of a misnomer. The fact that we can see any activity at all actually means we must have had a fairly large energetic effect, because it takes a fair amount of energy before you can even macroscopically see channel opening. And so here I'm just quantifying that. So here the free energy from closed to open for a wild type channel goes from something like maybe 3.7 kilocal per mole to minus 0.8 kilocal per mole. And the effect of ablating the hydrogen bond in this beta 2 subunit linker is about a third of that. And I want to highlight also that this is probably a lower level estimate, because our ability to estimate the free energy, the closed open free energy in the closed state for wild type, and is not very exact. And the way we've done this is just to look for very, very tiny blips of current that you might see when you apply PTX. And we came up with this number of like 0.2% open, which is very similar to the number that somebody else came up with using the same approach. However, I think that this is likely an upper bound, and the real actual free energy difference between closed and open in the resting state is probably even greater. So I think we're actually really starting somewhere over here in reality, which would mean that the effect of this mutation is even a larger fraction of the activation energy. Okay, so if this thing is coupled to the activation cycle, we would also expect allosterically to see an effect on the affinity of neurotransmitter at the binding site. So the reason that these channels open by neurotransmitter is the affinity for GABA is higher in open states versus closed states. And indeed, we see a left shift to higher sensitivity for GABA when we break this hydrogen bond. Okay, so up to this point, we know that this particular bond is super important to stabilize the closed state of the channel. If you break this bond in the beta 2 subunit, then it simply cannot stay closed. And energetically, we know it's coupled to the agonist binding sites, the GABA binding sites in the extracellular domain. But is it a part of the normal gating cycle? So we can't really tell that from structure. It's just it's just not obvious. So we turned to molecular dynamic simulations, and we did simulations in the antagonist-bound closed channel and in the GABA-bound desensitized channel. We also did simulations not shown here, but with the exact same result in a simulated open structure. And the take-home message is this: in the beta 2 subunit, what we see is in the closed state, the distance between donor and acceptor atoms is maybe about 3 angstroms. And that would correspond to a electrostatic hydrogen bond. And then when GABA binds and this transitions to a desensitized or open conformation, that distance is shifted to much longer distances, indicating that this bond is weakening or breaking. However, in the other 2 subunits, alpha and gamma, we basically don't really see much effect between the states. So it looks like there is only this state-dependent transition in this bond distance in the beta 2 subunits, and this nicely correlates with the experimental result that we only saw an effect on GABA activation when we ablated the bond in this beta 2 subunit, and we saw no effect when we ablated it in the alpha subunit. So in conclusion, we suspect that this bond is forming and breaking, or at least becoming closer and further apart in a state-dependent manner during the channel gating cycle. So in the closed state, this bond is closer, and it is necessary to keep the channel closed. And when the channel opens, these donor and acceptor atoms move further apart, and this bond is weakened. So this is almost like this molecular latch, if you like to think of it like that, between closed and open states. I would just, before moving on, say that looking at some of the new structures, it's not obvious that this bond is really changing distance. It also was not obvious between closed and desensitized states in the older structures as well. So either we're wrong in that this is state dependent. Clearly, we're certainly right that this is energetically required to remain closed. But my suspicion is that these structures just don't resolve these small effects, and we're likely going to need to turn to molecular dynamics simulation to get a better idea. And those simulations also suggest that this bond is not really locked into 2 distances. It's really dynamic, and that's probably why we don't resolve it well in these structures. It's moving all over the place with just a sort of mean shift in distance to closer in the closed state versus larger in the larger distances in the open state. So whether or not that remains true in simulations of the new structures remains to be seen. Okay, so this begs the question then. There are hydrogen bonds involving main chain atoms, all throughout the protein. Here we're really focused on these loops that couple the extracellular and transmembrane domains. But what about all these other bonds, main chain bonds? Are they important for gating? Here we found one that we know definitely is, but there's a lot of other ones. We've started to investigate some of these, and I'm just going to tell you about one of them for the rest of this talk. Which is this bond right here between a highly conserved tyrosine in the M2-M3 linker and so this is going to be a side chain to main chain interaction. And this is the main chain amide of a leucine in the canonical cis loop. And so this interaction looks like it could bridge these 2 loops. Once again, in some structures, this is identified with, and in others, it's not identified, primarily due to angular mismatch. But nonetheless, these 2 atoms are certainly in structural proximity. So we're going to use 2 approaches to ablate this. We're going to ablate this by removing this nitrogen using the alpha hydroxys, and we're going to ablate from the other side by doing conventional mutagenesis to remove this OH group. So here are the results in alpha-1, beta-2, gamma-2 receptors. If we, by ablating the bond by removing this oxygen. So here we're going to do a tyrosine to phenylalanine substitution, which essentially just removes this OH group. And what we see is in the beta subunit, we have a massive loss of ability to activate the channel. So this connection seems absolutely crucial to activate the channel efficiently. And you can only activate to about maybe 30% of normal if you've lost that bond. You also have a loss in sensitivity to GABA. Conversely, if you do the same mutation in alpha and gamma subunits, you see absolutely no effect. So this actually parallels what we saw with this other main chain hydrogen bond, that all of the effects seem to be happening in the beta subunit. And I would just point out that these two loops in beta subunit are sitting directly below the GABA binding site. So we're at the GABA binding interfaces. So it seems there's something very special about the contributions of these interactions at GABA binding versus non-binding interfaces. And here I'm just showing you the current recordings for that. So how do we actually know, let me just back up, that this is reduced in maximal efficacy? So we need some approximation of what 100% activation would look like. And the way we did that is we did GABA dose response curves and then followed with co-application of GABA and the anesthetic propofol. And this combination should activate the receptor presumably fully. And here you can see in the mutant that GABA is pretty much a weak agonist. It just can't activate the channel nearly as much, but we can still fully activate in the combination with propofol. Now, if we go to the other side, remove the nitrogen, we see exactly the same effect. So here we have the nonsense incorporation of the wild type amino acid, looks very similar to wild type, but then when we ablate it with the alpha hydroxy, we get exactly the same effect that we had from removing this OH group, a loss of sensitivity and a massive reduction in full activation by GABA. And so this, in my opinion, highly suggests that this bond indeed is the thing that is mediating this loss loss of this bond is indeed the thing that is mediating this loss of ability to activate by GABA. And here's just some current recordings showing the similarities. And finally, I just want to end by saying that this tyrosine is highly conserved in GABA and also in glycine receptors. And there are known mutations of this tyrosine that are associated with disease. So this would be epilepsy, developmental, intellectual disabilities, and in glycine receptor muscular disorders, hyperekplexia, which is a startle disease. And here, some of these mutants, so Y to C and Y to F, give exactly the same effect as we saw from either ablating the donor or the acceptor. I mean, that would be the F, but the cysteine mutant, which is also a disease variant, looks exactly the same. And I would point out that these, we've done this in both beta 3, which I'm showing here, and I previously showed beta 2. It's exactly the same. So this is conserved in beta 2 and beta 3 subunits across disease variants. And we have recently started to look at this in the glycine receptor, and this would be the exact same analogous tyrosine to phenylalanine. So removing the acceptor in glycine, exactly the same effect. And so loss of this hydrogen bond, I think, is very, very likely to be the atomic basis for the disease variants. So epilepsy, developmental disability, and hyperekplexia. And it is interesting, I would just say, that this tyrosine is actually not there in the cationic channels, only in the anionic channels, GABA and glycine. So there's something different going on in the cationic channels. It would be interesting to figure out what that is. But in GABA and glycine, this is a highly conserved, incredibly important feature for activating the channel. And it appears to only be important at the ligand binding interfaces, just like we observed previously for the within main chain hydrogen bond. And Then with that, let me just acknowledge the people who did the work. I should have said this as we were going along. I showed their pictures, the people who did each step along the way. So Cecilia Borghese did the vast majority of the work in my lab, and she was helped by Natrang Desai. So Cecilia was really the impetus behind this project. Also, some past lab members, Khadija and Pranavi, helped. Of course, doing the unnaturals, the alpha hydroxys, would have been impossible without Chris and Jason. And the molecular dynamic simulation were a collaboration with Eric Lindahl's lab, Rebecca Howard, and Samuel Eriksson, Nandan Holoi, and Yuxun Zhang. And Samuel did most of the work. And with that, I'd like to also acknowledge my funding and thank you for your attention, and I'll love to take questions.
Wonderful. Thanks, Marcel. That was a really fantastic talk. We are just about time, but I think we can do at least one question here. We have one from Emerson Carmona from the audience. His question is, what do the single channel currents look like, and how do you know the changes in macroscopic currents are produced by single channel conductance or open probability changes?
So we don't know. We haven't done single channel recordings. I think that would be a fun thing to do. What I would say is there's a lot of literature on single channel recordings of GABA receptor mutations and activations by various agonists and modulators. And the vast majority of these manipulations don't change single channel conductance. They only change the frequency of opening, so open probability. So the default assumption is that it is most likely a change in open probability and not single channel conductance. But without actually doing the recordings, I can't say with absolute certainty.
Great. Anything else? Any other questions from the audience? I'll just add one. I know this is a potentially really ignorant question, but maybe when you encode these alpha hydroxy amino acids, now you have an ester. Is there a concern for hydrolysis of the ester bond? I'm sure you've thought a lot about that. Is that ever a problem, and do you look for that? How does that work exactly?
So I mean, I haven't looked for that. I haven't been very concerned about it. I mean, I think Chris thinks that, these are pretty stable.
Okay,
What we do know is that I think if we somehow lost this bond, that the channel wouldn't function probably. So
right,
the
right.
fact that we have functional channels, I think, suggests everything is fine.
That's pretty good.
But we have not done anything specific to test for that.
Okay, cool. All right. Well, we are out of time. Thank you, Marcel and George, for two fantastic talks. This was really exciting, really fun to hear all your great work. And with that, I think we'll part ways and thanks again. All right. Take care, everybody.
Thank you, everyone.
Thank you.
Thanks.